Hey guys, good afternoon. We're going to start the last chapter, the chapter 20, and we have three sections in chapter 20. So today, part A, part B, and part C. Part A is the background knowledge of the reproduction, particularly the sexual reproduction. And part B is male reproductive system. Part C is female reproductive system. Okay. And if time allows, we will talk a little bit about the pregnancy. But the human sexuality and pregnancy, childbirth, those are not the part of our uh, the study here in human physiology. Okay, that is usually taught in health classes. So today, uh, I will go over sexual reproduction, how the in animals sexual reproduction is different from asexual reproduction, particularly in human being, how we compare them with each other. In human sexual reproduction, we will talk about sex determination. So what are the factors which determines whether the the newborn baby will be male or female, and what determines the development of accessory sex organs. So we will talk about male and female reproductive system organs, and then we will talk about accessory sex organs. Can you guys hear me?
I can hear you.
Yes.
Yes, I can hear you.
Okay, so somebody had Jessica, I think. She has some problem with sound. Can you hear now, Jessica? She's not responding. Okay. So we'll talk about the development of accessory sex organs, which is the part of the development of reproductive system organs. As you know, the hormone is the major neurochemical transmitter which control the reproduction as well as other metabolism of the body. So we're gonna talk about endocrine regulation of reproduction. What are the hormones which regulate our reproduction? Okay. And in that case, in that hormonal regulation of reproduction, we're going to talk about hormonal interaction. Several hormones have interaction, like hormones from hypothalamus, hormones from anterior pituitary, then hormone from gonads, how it interact with each other to maintain normal physiological function of the reproductive system. Puberty is the age at which secondary sexual characteristics and reproductive life of an individual starts. That is the puberty. And puberty is completely induced by hormones. So what is the role and what are the changes of in body? What is the role of hormones and what are the changes in the body during puberty? We'll talk about that. And we will touch base with human sexual response when they have during the copulation, which is the means of transferring sperm to the vagina. And in that case, there is physiological and psychological response are the change and response in the copulation. So we'll talk about some aspects of human sexual response in very brief. Okay. So let's just start with the reproduction first. So the main function of the reproductive system is reproduction, to reproduce. Okay. And when you reproduce, the organism in the universe or on the earth have two ways of reproduction: asexual reproduction and sexual reproduction. Asexual reproduction means you don't need both male and female. Only one organism can reproduce. For example, our bacteria. Bacteria is unicellular organism and they just divide into half and becomes two with equal number of chromosomes. And that gives another copy of that bacteria. That is asexual. But in sexual reproduction, when they mix gametes, they divide their chromosomes into half. And that's why there is need for haploid gametes to fuse together to make complete cell. Okay? And to do that, you need both male and female. And so when there is reproduction with both male and female, that is called sexual reproduction. The beauty of sexual reproduction is genetic variation. And you have done cell division, mitosis and meiosis. In the meiosis, in the prophase, there is crossing over, an assortment of the genetic component in the DNA, and they are mixing. So by meiosis in the ovary and testis, we divide cells and make a sperm and egg. There is like hundreds of thousands of trillions of genetic variables. That's why you never ever see an individual who is exactly the same with others. Whether somebody was in billions of years in the past or will be billions of years in the future, nobody will be exactly same to other individual due to that sexual reproduction and genetic variation. Clear? Okay. So before we go over the before we go the
I think Ryan had a question, Dr. Shaw.
okay, go ahead.
You wrote it in chat.
Oh yeah, that was me. I thought it was in the second phases of that's where the in meiosis is where the variation happened.
Crossing over in the metapage.
And well, because in meiosis you have two phases of everything, right? You have like the first phases of the regular stuff, and then the second phase. I thought it was like that's when all the variation of the genetics.
It is in the first page.
Okay.
So meiosis 1, that happens in our testis node. We're going to go there. It is in the first phase where crossing over happens. Second phase is just making two copy and divide.
Okay.
Yeah?
Okay. I had it backwards.
Okay. So the organs of the male and female reproductive organs, both male and female, has primary sex organs. In male, it is testis. In female, it is ovary. Testis and ovary mix the gametes. Testis makes the sperm, and the ovary makes egg. And then the sperm is transported by the other accessory organ, internal organs of the male reproductive system, like ductus deferens. Then there is several other accessory organs like which give the fluid and nutrition to the sperm, like seminal vesicles, ampulla of ductus deferens, ejaculatory duct, prostate gland, bulbourethral glands, and then there is penis through which urethra runs that transport the sperm to the vagina during the copulation. Okay. Other hand, in female, The ovary makes egg, and that is collected by fimbriae of the fallopian tube or uterine tube, which brings it to the uterus. But sperm pass from the vagina all the way. Okay, some of you have to turn off your mic because we hear side noise. Okay, so the sperm can pass from the vagina all the way to the fallopian tube where fertilization happens. And then fertilized egg comes to the uterus where it implants. From the uterus, they grow there, or egg usually die when there is no fertilization. So other organs of the female reproductive systems are like cervix, vagina, which is the birth canal, and then external genitalia are like the labia minora, labia majora, and the homologous to penis, there is clitoris. Okay, so these are the organs of the male and female reproductive system. And male is male because male has testes, and female is female because female has ovary. Because both testis and ovary makes hormones. Testis makes testosterone, and that creates male body. Ovary makes estrogen and progesterone, and that creates female body. Okay, so you will see these structures again and again. That's why I want to put it at the beginning. The PowerPoint I have posted in Canvas does not have it, so I'm going to remove that PowerPoint and put this one today. Okay, so life cycle of human. Okay, so adult male, adult female. Adult male testis makes sperm, and you know, adult female in the ovaries we make egg or ovum, and these both sperm and egg are haploid, means they are half cell. They have only 23 chromosome, not 23 pairs. They have only 23, only one pair of each chromosomes. Okay, now ovum has also, when these sperm and ovum during fertilization in the fallopian tube of the female reproductive organs, they meet together and fuse, that forms the full complete the zygote, and then the zygote moves from there to the uterus where they grows by mitosis and implant on the uterus. And then after 9 months you have again either male or female new human being. Okay, and that is now body with cells with 46 chromosomes. Okay, okay, so each zygote, which is the fertilized egg, inherits 23 chromosomes from mother and 23 from father. Okay, and that 23 and 23 makes 23 pairs. So they form like 23 pairs homologous chromosomes. They have each of them. Okay, out of those 23, let's see male, we have, whether we are male or female, we have 23 pair of homologous chromosomes in our cells. Take any cells, we have these 23 pairs. Of those 23 pairs of chromosomes, 22 of them are autosomal chromosomes. Those chromosomes that determines your hair color, your body weight, your body height, your facial feature, and other physical characteristic of your body. Whereas the 23rd pair, so if you number 1 to 23rd, the 23rd will have 6 chromosomes. That 23rd pair is 6 chromosomes. And if you are male, then you have XY chromosomes. If you see in the scanning, And determination of the size of the X and Y chromosomes. X chromosomes are bigger and Y is smaller. Okay. So in male it is XY, and in female it is XX female. So male is male because male has Y chromosomes, not the X. It is Y which makes X, makes the male. Okay, Y. So what is chromosomes now? Chromosomes are the length of thread where there is genetic code. It's the base pairs and the ribose sugar, and those base pairs are the genes codes. So the Y chromosome contains 80 genes, whereas X chromosome contains 1098 genes. What does that mean? That means it looks like X chromosome somehow in the process of evolution has changed and become smaller due to the deletion. And that's why you have lost almost 1000 genes in the X chromosome and it became Y. Okay. So it looks like the male was created from female. If you are believing in evolution mythology, then God seems created female first. And then from their gene, there is mutation and male become. Do you know animals, even in some animals kingdom today, these animals like some fishes, they can change their sex immediately from male to female and female to male. They can change. Okay. So if you are two XX chromosome and XY chromosomes, which looks like complete XX set or XY set?
XX.
XX. That's why complete female is complete human being and male lacks some gene. So let's see if X and Y, here is 1000 and here is only Y side is only 80. That means you are missing 1000 genes. So in male, the job of two pairs is done by only one. Let's see if you have any problem with a small site on this X chromosome, what happens? You'll miss completely. Let's say female lose some section, what happens? Female has similar copy of that in the left side here. So this side will protect female from that loss and still maintain the function. That's why there is very, very rare X-linked disease in female, and it is very common in male. Are you following me? Therefore, in males, approximately 1000 genes are only represented by a single allele from the X chromosomes. And that allele, if there is any single X allele, there is if any mutation, that will for sure cause a disease defect in male, and that is called X-linked disease. That's why there is large number of genetic disease caused by X-linked genes in male compared to female. Okay. Okay, so now sexual differentiation. So how when egg and a sperm. So when male makes a sperm, sperms are either X or Y. Okay. And if you have XX, that's female. If you have XY, that's male. But how and why X, Y differentiate into male and why X and X differentiate into female? What is the reasoning? Okay. So let's see here. Early embryonic gonads can become testes or ovaries. If you have Y chromosome, then it induces formation of testes. Do you know why? Because the Y has long arm and short arm. The a particular gene on the Y chromosome induces the embryonic gonad to become testis. So there is a small gene on the short arm of Y chromosome, and that gene is called SRY gene. And that SRY gene induces the formation of testosterone. Okay, if you have testosterone, then that embryo will develop into male fetus. If there is no testosterone, then that will develop into female. So it is presence or absence of testosterone which determines the sexual differentiation. You may have XY chromosome, but let's say your Y chromosome has no gene, SRY gene. In that case, even your genetic makeup is male, you will not be a male. Okay? In absence, so presence or absence of testosterone determines sex. If you have presence, male sex. If absence, female sex organs will develop. Okay? So let's see formation of testes and ovaries. So how we develop the primary sex organ of male and female as testes and ovaries. First 40 days after conception. Conception is, what is conception?
Like fertilization? Is it sex?
No, no. We did lab on pregnancy test.
The implantation.
Mhm.
Implantation, yes. Conception is implantation on the uterus wall. When fertilized egg implants on the uterus, that is called conception. Okay. So the first 40 days after conception, gonads of males and females are similar because we have not produced testosterone yet. That's why. Okay. Cells that will give rise to a sperm and egg migrate from yolk sac to develop gonads. So in the fetus, there is a sac, amniotic fluid, then there is germ layer, and there is a small sac on the side. That is called yolk sac. Yolk sac contains some cells, which are the stem cells for the gonads. And that stem cells then migrate from there and go to either ovary or the testis. Okay. Gonads have potential to become testis or ovary until testis determining factor. TDF causes conversion to testis. So now fertilized egg are in uterus and there is XY gene. On the Y chromosome, there is another gene. That gene is called SRY gene. That gene determines a protein. That protein is called testis determining factor. And that testis determining factor produced by SRY gene, what does it do? Produce and help to grow the testis. Okay. So here you go. This is the Y chromosome. It's Y chromosome. It has short arm. There is centromere where they connect both short and long arm. There is long arm. And then in short arm, you see there is a small blue region. That region contains SRY gene. This gene is Y-linked because it is found only in the Y chromosome. You will not find this gene on the X chromosome. So if you have, let's see, there is Y, a sperm, and ovum. Here is X sperm and X ovum. So you have XY and XX. When you have XY on the Y, short arm of the Y, you have which gene? SRY gene. SRY gene makes what? SRY gene goes to the cells and that genes makes the testis determining factor. Then testis determining factor start developing tubes, small tubes in the testis. That is called seminiferous tubules. And a week or two later, These testis determining factor also helps forming the cells outside the tubules, which is called extratubular cells, and that is interstitial cells or cells of the Leydig or Leydig cells. Now, seminiferous tubules wall has two kinds of cells: germinal cells and Sertoli cells. Germinal cells makes sperm, and Sertoli cells makes other hormones like inhibin and other. Okay. Interstitial cell, on the other hand, makes testosterone, and this testosterone hormone then further go to the cells of the embryo and further develop the structure of the male reproductive organs, and that's how male fetus develop. In absence of TDF or SRY gene, what happens? Zygote has no SRY gene, no TDF, so then rest of the structure in the female reproductive line will develop. So there is ovary developed, and then later the unit of the ovary, small unit, a small ovary within the ovary called follicles, that will develop later. Okay, so the summary is, it is Y chromosome. On the Y chromosome, it is SRY gene, and then SRY gene code for TDF, and that TDF makes the testis, and if there is no gene, then that makes the ovaries. That's how sex differentiation happens. But up to now, we are only internally male or female. The body feature later is determined by testosterone and the hormones from adrenal cortex, particularly zona reticularis of the adrenal cortex. Do you remember endocrine system now? We're going to use it, so remember them. Okay. Okay, so let's see, we have the Y chromosomes, and then we developed already seminiferous tubules. So seminiferous tubules, which produce sperm in adult, that appear within 43 to 50 days following the implantation. They contain germinal cells, which produce sperm around 42 days they appear. And then after that, certain times after like 2 weeks, then interstitial cells, which is called Leydig cells, that produce testosterone. Okay, and these appear like a cluster in the testis outside the tubules. Okay, in absence, if there is no TDF, then the same gonad, same structure, fetus develop the ovary in the line of female reproductive organs, and then later ovarian follicles develop like after 105 days. Okay, Leydig cells, what does Leydig cells do? So we have Leydig cells, germinal cells, and Sertoli cells. Sertoli cells and the germinal cells are in the wall of seminiferous tubules, small tubules in the testis, whereas Leydig cells are outside the tubules. Okay, so Leydig cells begin secreting testosterone at 8 weeks of development and peak at 12 to 14th week. That means very early stage, there is 8 weeks is like around 2 months. There is secretion of testis. So hormone, testosterone, are very high during the early fetus age, because that's the time you have to make sure that fetus is male. Okay, this high concentration during these 8 to 14 weeks masculinize embryonic structure of the XY chromosome zygote. And then after that, testosterone level declined till the puberty. So from early embryonic age till the puberty, there is no testosterone concentration high in our body. After like immediately before the birth. So testis is fully developed with the tubules and the interstitial cells, and it descend in the scrotum before birth, before the childbirth. Do you know why it has to come outside the body and get into the sac of a scrotum? Because the sperm formation occurs only in lower temperature, which is 3 degrees centigrade lower than the body. And if your testis is inside the abdominal cavity, both testis and kidney develop into abdominal cavity, and then testis come outside, kidney remain in the abdomen. Okay, that's why when testes descend down, they come with the peritoneum. And when we do histology of the testis in a human anatomy, you're going to see that. Okay, so that's why testis must come out of the abdominal cavity and hang in the sac of a scrotum. So you can cool down the testis 3 degrees centigrade lower than the body temperature where spermatogenesis occurs. Okay. If somehow in some newborn will not have testis in the scrotum, when the child is born, what do? We do suction, we check every pulse, vitals of the baby, and then immediately we palpate the scrotum if the male child and see testis is descended or not. If not, that's not good sign, because if you keep testis inside the abdomen, not in the scrotum for a long time, the body temperature will slowly kill down the sperm forming germinal cells, and that individual will be sterile for life. So we wait for descent by itself, and maximum I think we wait for a year. If within 1 year, if testis doesn't descend in the scrotum, that condition is called crypt orchidism. So crypt orchidism is undescended testis. And if it is more than a year longer, that means the testis is already dead. And if you don't remove, so after that, the treatment is surgery. You have to take out enucleation, take out the testis. If you don't do that, there is 100% chance that testis will turn into testicular cancer, which is very, very fatal. That's why you have to remove the undescended testis. Okay. Now we talked about how testis and ovaries developed, and now you're male and female. But to be complete male and female, what you need to compliant with your primary sex organs. And to that, you have male accessory sex organs and female sex organs, accessory sex organ, which must match with the primary sex organ. So male accessory sex organs are derived from Olffian duct, and female accessory sex organ derived from Mullerian duct. So here in the diagram, you can see this is sexually indifferent stage around 6 weeks. So you have here, you have primitive gonads, and then you form the duct around it in the fetus. The blue duct, which is medial, is Olffian or mesonephric duct. And the red one here is the Mullerian or paramesonephric. On the parallel side, on the side of mesonephric is paramesonephric duct. So if the primitive gonad has TDF, then they will produce testis determining factor and testosterone. And in presence of testosterone and Sertoli cells, you remember the seminiferous tubules have Sertoli cells. Okay. So Sertoli cells will produce another thing that is called MIF, Mullerian inhibiting factor. And that will inhibit these Mullerian duct. If you inhibit Mullerian duct, what happens? no development of female accessory organs like fallopian tubes, uterus, and upper part of the vagina. The blue structure will develop into epididymis, vas deferens, seminal vesicle, ejaculatory duct, which are the part of male accessory sex organs. Okay, so here you go. There is indifferent gonads at around 6 to 8 weeks. You have testis determining factor with XY chromosome when there is male fetus, then testis makes testosterone and Sertoli cells. You see, at about 60 days, Sertoli cells secrete another protein called Mullerian inhibiting factor, MIF, and that inhibit the Mullerian duct, and Mullerian duct regress, degenerate, and once Mullerian duct or paramesonephric duct degenerates, that means this side will not develop. So what will develop then? If you have MIF, everything mesonephric duct or Wolffian duct will develop under the influence of testosterone into epididymis, ductus deferens, ejaculatory duct, and then the prostate gland, penis, scrotum from the other structure. If you have no SRY gene and no MIF, then all the structure of female accessory organs develop like uterus, uterine tubes, vagina, labia minora, clitoris, labia majora, these structures. Okay. So you can see here, this is the diagrammatic view. Like when you see in early stage, both male and female look same. You see here, there is genital tubercle, then there is labio-scrotal swelling. This is swelling for the labia and swelling for the scrotum. And there is cloacal membrane, urethral fold. Okay, so you see the color here, you match here. Let's go here. This side we are developing male, and this side there is female. This side is under the influence of SRY gene and testosterone, no testosterone, and no SRY gene. So female development. So let's see here. The genital tubercle in male will develop glans penis and phallus, which is the penis, and head of the penis, glans penis. If there is no testosterone, then same genital tubercle will develop into glans clitoris. Whereas the urethral fold and labioscrotal fold, you see, labioscrotal fold in male is developing into scrotum, whereas the labioscrotal fold in female is developing into labia majora, and urethral fold is developing into labia minora. The genital tubercle ultimately is developing into glans clitoris. That's why glans penis and glans clitoris in male and female are homologous structure. So let's see if a female is bodybuilder and try, let's see, to build the muscle, start taking the synthetic testosterone. What will happen? That female will develop the enlarged clitoris like penis. There is certain disease like polycystic ovarian disease. In that case, the female ovary cysts cells start making some male type hormone, and they act on these clitoris, and there is clitoromegaly, enlarged clitoris in those female, body here, male pattern body here, and that's how you can see the male pattern change in female. Okay. So in the absence of testosterone, clitoris forms instead of penis and labia instead of scrotum. And these two structures are homologous to each other. Okay, but is there always binary? Like either you are male or female? No. Sometimes you can have XX, or that extra X or absence of X. You have X, X, Y, or you have only X and no Y. And in that case, you have intersex. Or sometimes you have normal XX or XY, and then you need hormone. And to make hormone, you remember in the endocrine system, there is several enzymes which is essential for conversion of cholesterol to hormones. And then if there is any deficiency of enzyme, then you will not make that hormone, or you will make more of that hormone. In that case also, you will have unmatching accessory sex organ and external genitalia, and that condition is called intersex. Okay. And there is like variety in nature, in other animals as well as in human being. Okay. So when you have both male and female in one body, then we call them hermaphroditism. Okay, that is called hermaphroditism. In plant life, in animal, in organism, other organism, there is a lot of hermaphrodites. They have both male and female in same body. But it can happen in male and in human. And in human, it is usually due to the difference, an abnormality with the hormone deficiency, or the receptor insensitivity or lack of the receptor, or genetic abnormalities. Okay. So I'm not going to go in detail because that is another chapter to enjoy. You guys can go and check it. So pseudo hermaphrodites can be true hermaphrodites or false hermaphrodites. True hermaphrodites means if you have both male, full male and female, you have both testis and ovary, functioning testis and ovary in one male, one person. That is called true hermaphrodites, which is pretty much not found in human being. Whereas pseudo hermaphrodites are common in human. So pseudo means false hermaphrodites have either testis or ovaries, but have accessory organs and external genitalia that are incompletely developed or inappropriate. Inappropriate means if you are looking like male and you have not the penis and other structure looking like male, that is inappropriate. If you're female and your clitoris is enlarged like penis, that is inappropriate. You are male, but you have breast enlargement like female, that is inappropriate. You are female, but you have external body hair, like all facial hair and other body type, like male, that is inappropriate. So these unmatching structures, if you have, then we call them pseudohermaphrodites. Okay, there can be male pseudohermaphrodites or female pseudohermaphrodites. So most common cause of female pseudohermaphroditism is congenital adrenal hyperplasia. What does that mean? So by birth, if your adrenal gland, let's say you are female, but your adrenal gland, zona reticularis, is producing a lot of sex steroid similar to testosterone. What will that do? That will change external genitalia and external body feature to female like male, and that is called female pseudohermaphrodites. In males, one cause is testicular feminizing syndrome. So what happens? Let's see, you have XY chromosomes, you have XY chromosomes, and you have Y gene, you have everything. You are making testosterone, but any hormone needs target cells, the cells which have receptor for that particular hormone. So you have testosterone, but you don't have receptor for testosterone. Will that testosterone work? No. And if testosterone is not working, will you develop the function of testosterone structure in the body, like enlargement of the penis, the body feature, body mass, everything? No. And that is called testicular feminizing syndrome. So in testicular feminizing syndrome, testes are normal, but there are no receptor for the testosterone. So what happens to the individual? Individual develop very female appearance, but is infertile. Okay. Inside they are like male, but outside they will have totally female like behavior. And that is one example of intersex too. And there is several other stuff in and so debatable a lot of time, and there is a lot of things biological. Sometimes it is nothing you can see. There is like anything in the male body, complete male, everything male structure, but the neurons receptors for testosterone is different, and you will not respond the same way like other male respond. So these are the several intersex conditions. This is,
Professor,
yes.
if you catch any of these things in children at an early enough age, is there anything you can do to prevent like it happen continuing?
You're gonna get into trouble.
Okay, if it's too hard of a question, that's okay. I just was curious.
You can get into trouble because it is hard to do it. It is very hard to do it. Like how you're gonna do, you can do therapy, testosterone therapy, but whether it is appropriate or not, it is social acceptance too. So those things are like iffy, but in some condition you can change the whole body from female to male to female. Why you cannot do that? You can do that, but that's debatable.
Yeah, but
Sorry, I guess just more in terms of like hormones and stuff, but you
Yeah.
answered my question.
Okay.
but having the absence of these hormones or genitalia, it doesn't really affect your overall health in general, does it?
Yeah. So the hormones, like if you have complete, this is like you have testosterone. Testosterone has not only receptor in the genitalia, it has receptor on your muscle cell, in your fat cell, in your brain cells. So if you have receptor in the brain cells, you will still think like male. But if you have not that receptor in your other body part, then you will not. Like if you have not in the muscle cell, then you will not develop muscle mass. So this is like which receptors are modified. And a lot of things we don't know completely too. Okay. Did I answer your question?
Yes.
Okay. Okay, endocrine regulation. Oh my God, we are done. Time. Okay, next thing we're going to talk about, we have 4 days lecture, so we're going to cover in next class. Okay. Thank you guys.
Professor Shea, can I ask a quick question? It's going back to a couple slides back where you talked about the gonads before the sex is determined. We hear a lot about artificial sex determination of a child. Do they then apply the determining factor at that point before they develop either male and female if somebody wants to make sure they get a certain gender for their child?
No,
Yeah, this slide.
gender is like at
Oh,
55 days, you are already determined gender.
Oh, okay.
Yeah.
Okay. All right.
Did
Thank you.
I think I did not completely heard your question. Can you say it again?
I was more on this slide where at some point
Which
the
slide is
gonads.
this? Which slide is this?
It was the one you had previously. I think it was slide number.
Here?
Yes, actually, the one before works. Yeah, either 11 or
12?
or 11 or 12. Yes. So I talked about how the external genitalia is the same, and then the secretion comes in the determining factor, and then that's when see the development of these different genitalia. We hear a lot about like artificial sex determination of a child. What is the physiology behind that? How do they apply that at what stage?
So how, your question is how
Yeah.
the genitalia is determined?
No, how people can determine the sex of their children, and maybe that's more like a kind of like an artificial determination. People say,
Or
"I want to have a
in
child."
early
Yeah.
age, the only way you can determine is like nobody wants to do the supra, what is that called? Pubes. It is, you take the amniotic fluid and do chromosomal analysis, which is
Mm.
invasive, you don't do. So the most important way is non-invasive, which is ultrasonogram.
Mm.
And by the 12 week, you will have this phallus development already.
Hmm.
And based on that, they can determine the sex.
Okay.
Yep.
All right. Well, thank you.
Thank you.
I have a question. When you were talking about cryptocordism,
Crypt or cordism.
cryptocordism.
Okay, that's fine.
When the testicles are undescended and they do surgery, do they have to completely remove it, or are they able to put it into the? Are they able to help descend it into the sac, or do they have to completely remove it out of it?
No, there is no manual pulling and putting into the scrotum. They remove it completely because I said there is 100% chance that the testicular tissue will develop into cancer.
Oh, I see. Okay. And they don't have any like there's no like other alternatives for um putting testicles in like I don't know how to explain it, but like other alternatives for male to produce sperm in an
No,
with
if you have no testis, then there is no, like you have killed all the cells. If you are like 1 year more
mm-hmm.
in the
An
abdominal gland.
artificial, like an artificial way to produce it
No,
for men. There's no.
no. Neither egg or not a sperm. Nobody can do that.
Not yet.
Not yet.
Okay, well thank
Yeah.
you, Professor.
Okay. Thank you guys.
Thank you.
Thank you.
Thank you.
Thank you.
Bye-bye.
Thank you, Professor.
Thank you.